Welcome to another session on biomedical instrumentation. So in our last session, we had a very detailed introduction into biomedical instrumentation. We had discussed on why biomedical instrumentation, the application of biomedical instrumentation, and the various features required by the instrument system for acquiring biomedical signals were understood. So in today's session, we will look more into where exactly to find biopotentials and what are the concepts behind such a biomedical signal. We have already understood that the sources of biomedical signals include bioelectric signals, bioacoustic signals, biomechanical signals, biochemical signals, biomagnetic signals, biooptical signals, bioimpedance signals, to name a few. Now, to carry out certain functions in certain systems of the body, they generate their own monitoring signals which actually convey useful information about the function they represent. Now, these biopotentials are actually associated with nerve conduction, brain activity, heartbeat, muscle activity, and so on, to name a few. Now, the biopotentials are actually ionic voltages produced as a result of electrochemical activity of certain special types of cells. And these signals can actually be converted by suitable transducers into electrical signals. They can be acquired, measured, and displayed for further diagnosis and treatment of various diseases. Now, this idea of electricity being generated in the body dates back to the 17th century, when an Italian anatomy professor, Luigi Galvani, claimed to have found electricity in the muscle of a frog cell. Later, in 1903, the Dutch physician William Einthoven introduced the string galvanometer, which would actually be used for any practical application for extracting these potentials. And later on, with the development in solid state technology, it was possible to acquire these signals in a better way. So, as I said, certain cells in the body generate these bioelectric signals because of certain functions and certain activities. Now, the basic concepts of the origin of bioelectric signals actually starts with a volt. A volt is nothing but a charge difference between 2 points in space. Now, going deep down to a cell actually has an intracellular structure and certain constituents, and similarly, surrounding the cells we have the extracellular fluid. So the intracellular and the extracellular constituents of a cell can be explained with the help of ions or charged particles. So we have the concentration of negatively charged particles or anions, which include large intracellular proteins and chloride ions, while the cations, which are positively charged particles, are having major constituents as sodium and potassium. Now at the membrane of a cell, as I have already pointed out, inside we have the intracellular fluid and outside we have the extracellular fluid. The boundary between them is the cell membrane. Now this cell membrane has a selective permeability property whereby certain ions are allowed into the cell while certain others are blocked. So accordingly, in this condition, the inside of a cell membrane is mainly constituting the anions, while the outside is constituting the sodium ions, the cations. The reason because is the membrane is permeable to potassium ions while it is semi-permeable to sodium ions, which means that it blocks the entry of sodium ions and permits the entry of potassium ions. So as a result of which, in a resting phase of a cell, there is a heavy concentration of the cations, that is the sodium ions, and the inside of the cell has a heavy concentration of the anions. As a result of which, the resting phase of a cell is positive outside and negative inside. So this is how it appears. And it has been pointed out that the membrane potentials in various cells ranges from about -60 to -100 millivolts. Now under this condition, a cell is said to be in its resting state and is said to be polarized. Now the boundary of a cell in its polarized state is found to be so because it is found that the permeability of potassium is 50 to 100 times larger than the permeability to sodium. So this leads to a constant potential difference across the resting cell membrane. So this is the picture at the boundary of a cell membrane. So we can see that you have an extracellular region, an intracellular region, and the boundary. Now this boundary is also fitted with channels so that there is a possibility, if there is a possibility of movement of ions into the cell, these channels will open up. And so this is another animated picture showing the condition at the cell membrane. So having a meter and measuring the potential across the cell, we find that it is about -70 millivolts. Now this is -70 because we are measuring it from inside to the outside of a cell. Now at this junction, if some external energy is applied, the membrane changes its characteristics and begins to allow some of the sodium ions to enter. Now this movement is actually helped with the help of or with the aid of the sodium channel. This movement of sodium ions into the cell constitutes an ionic current flow that further reduces the barrier of the membrane to sodium ions. The net effect is that there is a heavy influx of sodium ions into the cell. The ions actually rush into the cell to try to reach a balance with the ions outside. At the same time, the potassium ions, which are higher in concentration inside the cell during the resting state, try to leave the cell but are not as fast as the sodium ions. as a result of which the cell has a slightly positive potential on the inside due to the imbalance of the potassium ions. Now, once again, if we measure the potential between the inside and the outside of the cell, we find that the potential is now approximately plus 20 millivolts. Now, this is said to be the action potential of a cell, and the cell is said to be in a state of depolarization. However, this depolarization will not remain so for a longer period. The cell membrane actually reverses its normal polarity after a brief period before re-establishing the resting potential. So this is observed in this graphical representation. So we can find that the membrane potential on the y-axis and the time on the x-axis shows the whole picture of the activity. So initially it is at the resting condition, which is minus 70 millivolts. Now, due to certain activity or excitation at the boundary of the cell, there is depolarization, as a result of which there is an action potential. Now, this action potential is expressed as plus 30 millivolts. So where we find that there is a heavy rush of sodium ions and a slow rush of potassium cells outside, so whereby the polarity of the cell reverses. However, this stage is not permanent. There is a process what is known as repolarization, whereby the cell will get back to its normal state of resting potential. So what is it that happens at this condition? So this is the different phases of cell, where we find that the resting phase, the action potential, and again the repolarization phase, and coming back to the original condition. So during the repolarization, the sodium-potassium pump will be very active. So what happens is, the sodium pump quickly exports the sodium ions through this pump, and the cell again becomes polarized and assumes its resting potential. And this activity is what happens at repolarization. Now this activity happens with the help of high energy phosphate content, which is called as the ATP. So we'll just observe the animation, what happens at the boundary during repolarization. So the sodium ions get attached to the pump, while the potassium ions from the outside is transported into the inside of the cell. And this happens with the high energy phosphate component. So the pump opens to transport the sodium outside, and the potassium meanwhile attaches so that it can be reintroduced into the cell. So once again we can see the graphical representation which shows the activity very clearly. So the resting phase, because of some external energy, the action potentials generated, which is peaked, and finally this stage is not permanent, and during repolarization, using the pump, the sodium is again brought out and the potassium is brought inside, thereby the resting potential is again reconstituted. There may be chances that the potentials may not be brought to the original condition instantaneously, but there may be a few after potentials during which this resting phase is reinstated inside a cell. And all these potentials can be actually measured with the help of micropipettes, that is a hair-like pipettes, hair-like electrodes, which are actually used for measuring these potentials without damaging the activity of the cell. Now the action potentials actually depend on the type of cell that is producing them. We find them very quick in nerve and muscle cells, where the repolarization is also quick after the depolarization. So action potentials may appear as spikes of just 1 millisecond of total duration. However, we find that these activities are very slow in heart muscles. So the action potentials are found to last to about 150 to 300 milliseconds. Now for the brief time during which the cell does not respond to a new stimulus is called as the refractory period. Now this is due to the after potentials that follow an action potential, as I said earlier. So after the action potential, during the repolarization, the cell may not be in the original resting state, but it may have a few after potentials, and this leads to the refractory period. And one more aspect which has to be understood about the action potential is the rate at which the action potentials propagate from cell to cell. And this is known as the propagation rate or the conduction velocity. Now the propagation rate are different for different types of cells. For nerve cells, it is found to be 20 to 140 meters per second, while for heart muscles, certain cells it is 0.2 to 0.4 meters per second, but certain other delay fibers in the heart muscles may also introduce a conduction velocity at a very slow rate of 0.03 to 0.05 meters per second. I conclude this session with this slide. We'll understand more details of the action potentials and the biopotential of a cardiovascular system in our next session.